You'll see for this microscopy lab that I don't have my garb. For us, this is mostly going to be a dry lab. Now, what you're going to want to do is open your virtual microscope. If you go to that little box in the upper left-hand corner of your hands-on lab screen, there's a little arrow. That arrow opens up the drop box that has not only your materials and access to your gradebook, but the last link there should be to the virtual microscope. So go ahead and get that open before starting this lab. Now, what you are going to notice is that the microscopy lab is set up for us to have a real microscope. And there was the option of renting a microscope, but that was going to add almost another $100 to your kit. So at Rock Valley, we decided we didn't want to add that to your kit. So we're going to get some of the experience of using a light microscope. Now, most of us groan about using a light microscope, and that's unfortunate because really, when this was invented, it was a huge technological advancement. People could finally peer inside of cells, and it helped people to correct their misconceptions. There used to be misconceptions about what was happening inside of cells, like the idea that inside of the sperm and the egg, the gamete cells, there were entire whole little organisms all crunched up inside of there. And that all they had to do after fertilization was just expand like a big balloon. We now know that that's not the case, but they didn't know any better. They couldn't see inside of a cell. So when you are doing the microscopy lab, one of the first things you're going to notice is that it's going to ask you to calculate the field of view. We can't do that because we don't have a microscope or a ruler. So I want you to leave the data spots blank for calculating the field of view. But I do want you to answer the question about why calculating the field of view is important. You should be able to answer that from the exploration section of the hands-on lab. The other thing that I wanted to give you a heads up on is in your first set of experiments, the lab is going to ask you to look at the letter E and look at colored threads. So when you have your virtual microscope open, just go up to your slide library, and what I was doing was typing into the slide search bar, letter E. You would bring up that slide, and then I could focus it, take a picture, and upload it, just as if I had my own microscope here at home. So follow through the lab, go ahead and look up those slides in your slide library, take your photos and upload them, just as you would if you had your own microscope at home. For the last lab, you're going to be asked to do a wet mount. Now hopefully with your lab kit, you did receive those slides and the cover slip. And though it seems kind of strange to go ahead and do the wet mount, please practice that at home. Upload the picture with your name and the date of the wet mount slides that you make of your own cheek cells. Just be careful when you're scraping your cheek, trying to get those cheek cells off for making the wet mount. Don't draw any blood. So go ahead and do those as if we were to look at them underneath the microscope. And the reason why I want you to do this is because if you are going further in biology, people are going to have the expectation that you know how to do a wet mount. They're just going to say to you, make a wet mount of the following things. And then they will move on, and you're going to be left there in the dark. So I want to make sure that you get that experience so that wherever you're going, you don't have that awful feeling of not knowing what to do. So go ahead and make those 2 wet mount slides, and then go to your slide library for the cheek cells, both stained and unstained, and use those slides from the slide library to finish out the rest of the lab. In other words, you will be able to complete all of the lab using your slide library, using that virtual microscope, and the slides and cover slips for the wet mount that came with your kit. The only part that you will be unable to do is to enter the data for calculating the field of view. Hopefully this clears up what you need to do for this lab, and I will see you guys in the next lab with my goggles, my gloves, and my apron. We'll get back to doing some of those more wet labs that we're accustomed to in our next lab. As always, email me if you have any questions.